Also, will it be locked on this screen or will it share video when people are talking?
Right now it's locked on this screen. Did you see it change for you when you spoke? Because I keep seeing the main screen. Okay, yeah.
I see the main screen.
Do you want me to unshare it so you can see your video first?
Nope, that's okay. Thank you, Sarah.
I don't know if you guys could hear that, but that was my yellow lab in the background there, working away. Still see we've got a number of attendees streaming into the call, so we'll give it a few more minutes here, everyone, and then we'll be started. Okay, I think we'll go ahead and get started here, everyone. Good morning and good afternoon. My name is Hayden Bliss. I'm the Director of Sales and Marketing here at Biomark, and wanted to thank everybody for joining us this morning. I hope you are all safe and healthy and your families are safe and healthy as well during this challenging time. I was looking over the attendee list this morning, and we've got a great crowd here with us today. People attending from Florida, Utah, Wisconsin, Oregon, Washington, our home state of Idaho, Montana, California, and we even have some friends across the pond joining us from Ireland, Spain, and Italy today. So we've got a great webinar coming up for you guys today. This is going to be put on by our Applied Biological Services division here at Biomark. These webinars are an effort for us to reach out at this time, as many of us are working remotely. So this is our effort to stay in touch with you all, engage with you. A quick update on Biomark. We are currently open still. The majority of our staff are working from home. As you can see, I'm hanging out here in my family's little pop-up camper. The garage was, it was a little too easy for my 4-year-old and 2-year-old to come into the garage and distract me. So I moved out to the camper. As many of us know, there's new challenges that come from working remotely. So we appreciate and we understand those. Or we do have our production staff and our shipping staff still working at our facilities, and we're taking the necessary protocols and procedures to make sure that they are safe and healthy. Our goal is to stay open and to be able to continue to provide products and services to you all. So we are here at this time, so please feel free to reach out to us if you need us. A few housekeeping items and then we'll begin on this webinar. This will be recorded, So if you, and we'll put it up on our website and on social media when this is done. So if you want to go back and review something or share it with anybody, by all means, please feel free to do so. We are in listen only mode, as you saw on the sidebar on the right-hand side of your screen. This helps us, in case somebody has any microphone or feedback issues. That way you can always hear us that are presenting. And so if you have questions, please put them in the sidebar there and the moderator will ask them in real time. So I encourage you to ask questions as we're going. This will help hopefully answer everybody else's questions and maybe spur some other questions. We will also stay on the call afterwards for a while as questions continue to come in if anybody wants to engage in further discussion. So please feel free to do so and we'll hang out as long as everybody does. So with that, I will introduce my colleague, Chris Beasley, who is our Director of Applied Biological Services at Biomark. Chris, take it away.
Thanks, Hayden. I appreciate it. And thanks to all of you for joining the webinar today. I hope that you and your loved ones and colleagues are all safe and healthy. Just want to say that your participation today sort of underscores my respect for our community. I know that all of you continue to advance the state of science despite all of these challenges. That being said, I sincerely miss the free and open discussion that we have at conferences. I think at the end of the day, all of us recognize that our work benefits when we present it to our colleagues, and that is ultimately the goal of this webinar, to share ideas, openly discuss their merit, and advance the state of the science. So with that being said, this is a little bit different than a standard presentation. I hope that you guys will feel free to use the chat feature to ask questions during the presentation. I'll try to moderate those and pass them along as efficiently as possible to the speakers. So if I fail to ask a question or you just want to stick around and elaborate on content, we're all going to stick around on the phone after the formal end of the webinar, and I'd welcome you to join us. So without further delay, I'd like to introduce Sarah Hoffman, Richie Carmichael, and Kevin C., members of the Applied Biological Services Division here at Biomark. So take it away, Sarah.
Thanks, Chris, and good morning, everybody. Today we were talking a little bit about the approaches that we've used to pair fish to habitat and how we are collecting data, analyzing data, and then rolling that out for implementation. And the majority of our work has occurred in the Columbia River Basin here in the Pacific Northwest United States. And a lot of the work that we are currently doing is on endangered Chinook salmon and steelhead. But most of us have some pretty diverse backgrounds. So a lot of the tools that we are developing, we're working on with an open mind as far as applying them in different ecosystems and to different species. So for those of you who aren't familiar with the Columbia River Basin, again, we're located here in the Pacific Northwest of the United States. And one of the challenges, as you can see from this white outline of the basin, is obviously a very large spatial scale, as well as multiple states, countries, and provinces. So there are quite a few coordination efforts happening in place as we are looking towards these efforts for long-term status and trend monitoring of these salmonid populations. And so the three major topics that we are going to be exploring today are all aimed at optimizing the efficiency of data collection analysis and then also extrapolation throughout the basin. So this morning I'm going to talk a little bit about how we are working to optimize our data collection, and then Richie and Kevin are going to jump in and talk more about the analyses and building a framework that leverages all of the available data out there. So I'm going to start with some of the efforts that we have made to increase our efficiency as we work towards data collection. And this really comes in the form of a protocol that we've been working on for a number of years called the Drone Assisted Stream Habitat, or DASH protocol. And essentially what this does is use multiple data sources paired together temporally and spatially in order to avoid site level data extrapolation that we have used in the past. So we still have the ability to ground truth some of these data with on the ground surveys, but we are able to extrapolate in hopefully a more effective way. And the primary imperative behind rolling out this protocol was to find a way to have these scalable data collection and analysis efforts starting at the channel unit or reach scale and working all the way up to watershed. So it allows us to perform a scalable effort given the needs of the project. And all of this is used to make these data-based prioritization and implementation suggestions for the groups that we are working with. So at the most detailed level of this protocol, we are going out and conducting these rapid on-the-ground surveys. And the idea here is that we are able to move through a river system relatively quickly, but still collect the fine scale data that we are looking for. And so we are moving through these reaches and delineating channel units as we go, and collecting habitat metrics like you can see Richie here measuring undercut characteristics, or things like cover, substrate, your traditional river habitat metrics that were initially based on the Columbia Habitat Monitoring Program, or CHAMP. So trying to leverage that legacy data and find ways to pair that into the new data that we are collecting. And we see characteristics, or this type of data collection will be used or relied on more heavily when we are interested in that added level of detail at something like a restoration site, where we want to be able to go in and give quantitative estimates of the change in habitat, and then also measure directly any uplift that may have occurred. The next level of detail up would be drone surveys, which can be, occur at the reach or watershed level. And it's a great way to safely, quickly, and cost efficiently collect large amounts of high quality data. And some of the products that come out of that are high resolution RGB imagery, from which you can produce these geo-referenced and ortho-rectified tiled images. So for example, in this figure, everything that you see that is white, so this snowy landscape, is all drone imagery that's just been overlaid onto satellite imagery. So it allows you to cover a rapid amount, or excuse me, a large spatial scale in a rapid amount of time. And from those data, you are getting sometimes sub 5 centimeter pixel resolution. So you're able to zoom in and still get a high quality amount of data within that imagery.
Hey, Sarah, can I interject for a second? We had a question come in. A couple of people were asking what type of drone you're flying, what altitude, and some questions about how much area you can actually cover, like in an hour or a day.
Okay. So we are flying we have two drones, a DJI Phantom 4 with an RGB camera, and then we also have a DJI M200 that we are flying with a multi-spec MicaSense Ultim. And Richie, do you think you can tackle the area and altitude questions?
Yeah, so typically we fly at 50 meters, which produces around a 2.5 centimeter ground sampling or pixel size. And I would say in an hour, we're covering roughly 5 kilometers of stream, which includes the floodplain as well. Typically, we only fly for about an hour because we try to fly during solar noon, which is more optimal conditions, so the sun's at its highest point to reduce shadows. And we try to fly in constant light conditions as well, so either fully cloud cover days or full sunshine days.
Excellent. And a couple other questions came in about what sort of software you're using, both to deal with post-processing and to develop visualizations like we're looking at here.
Yeah, I can take that one. So currently we're using Agisoft Metashape to scale and ortho-rectify and mosaic our images, but that also allows us to apply some photogrammetric techniques, so we can go from 2 to 3D imagery pretty seamlessly in Metashape. And then we use QGIS and ArcMap to create a lot of our visualizations, but we also create a lot of our visualizations in R and Python as well when we're trying to automate some of those mapping capabilities for reports and things like that.
Thanks, Richie. And that's a really good transition into, as Richie mentioned, there is a three-dimensional component. So to get at that third dimension, these softwares are allowing us to go in and produce a high resolution dense cloud, or something like a digital elevation model where we can look at more than just the flat imagery. So that's another advantage to using the drone moving forward. And this is just an example of what one of those dense cloud fly-throughs looks like. Oh, hopefully my computer is going to play it. Well, maybe not. But you can produce these high-resolution fly-throughs that allow you to take a virtual tour of your site in 3 dimensions, which is a great advantage when we're working with a number of different stakeholders from different backgrounds. And then the third level of detail or scale out would be leveraging any of the publicly available remote sensing data. So in the form of satellite imagery or the ability to incorporate things like LiDAR into our analyses are something that we are working towards right now. And the great thing about satellite imagery or any of these publicly available remotely sensed data is that they're almost always being updated. So you have this continuous near real-time data source for free, and incorporating that into the work that we are doing allows us an added layer of detail and monitoring in order to help us on these broader watershed scale projects.
Sarah just had a question come in asking where people can obtain satellite imagery and whether it truly typically is free.
Yeah, that's a good question. And Richie, I'll probably ask you to jump in on the end of this, but a really good tool that we have used is the Google Earth Engine. And that is a free repository for all of these remotely sensed data. So generally in the form of rasters, and it can be anything from just the straight imagery to things like NDVI or annual precipitation, temperature, things like that. And it is free. All you need to do is register as a Google developer, which is another free process that allows you to access all of that data repository. Richie, is there anything you want to add?
The only other data source I would add is the NAPE imagery data source that we utilize quite a bit. So typically those are flown every 4 years. And I believe you can access that through the USGS data portal. That's only available in the US. So folks in Europe, or outside of the US, I'm not exactly sure what your best source would be, probably the Google Earth Engine.
Yeah, that's a good point, Richie. And I didn't think about that, but a lot of the government entities have these data up on their website. So for example, I work primarily in the ocean, and I know that NOAA and NIMS have a lot of their data posted to their website. So with a little bit of scouring, you can find a wealth of data that are out there.
Yeah, and there certainly are private, you can pay to get higher resolution imagery from groups that have sent private satellites up, but for the most part, it's all free and freely available.
Okay, and so another added layer of detail that we are incorporating in the past year has been the use of a multi-spec camera. So again, we're flying with a MicaSense Ultim, which is a 6-band camera. So we have those 3 visible layers, which are the red, green, blue bands, in addition to a long wave infrared, which is a thermal band, what you see here, as well as a near infrared and red edge. And what those additional bands are giving us is added data, so something like NDVI that you can pull directly from the imagery. And where we see this being particularly useful is as we are developing these automated post-processing tools to do things like pull out all of the water from the image, having these added bands has significantly helped that process. So it's creating an added layer of differentiation. So in this image, for example, you can see a much starker difference in pixel value between the river in the thermal image as compared to the RGB. So these added layers of data are just helping us with these processing tools as we are moving forward. And so what we have been developing, and this is really Richie's baby, something that he's been working on for a long time, is a supervised random forest classifier to pull out these different types of cover or different land types throughout an image. So for example, in this image, you can see that every pixel that is water is being colored blue, vegetation green, bare earth gray, and then the woody debris is brown. So this is a pretty large scale image here, and you can see that the supervised random forest pixel classifier has done a pretty good job of pulling out those different types of cover. And if we zoom in, you can see on a much more detailed view that the same thing is true for these woody images. So for example, everything that is being highlighted in red here is being considered a wood pixel. And we know that there is definitely a trade-off between using something like this image classifier and actually going out into the field and counting and measuring every single one of these woody pieces. But it is definitely a trade-off in labor and time and cost to be able to feed a quick drone flight into these processing tools and pull out a good estimate with some measure of uncertainty of how much wood, for example, is in your stream as opposed to multiple days of survey effort.
Hey, Sarah, wanted to jump in here. We got several questions about this. The first deals with what programs you use to develop this random forest classifier and how you trained it. So probably questions for Richie.
Yeah, I can take that one. So the random forest classifier was developed in R, which is an open source computing language. And then we train it by placing random points through the image. And those points then extract pixel values throughout the image, and then we manually classify each one of those pixels. So for example, if a pixel falls on a piece of wood, then we will label that pixel or that point wood. If it falls in vegetation, we would label it vegetation, and that's what we utilize for our training data set. So essentially each point extracts, in this case, 6 values, which relate back to those 6 bands that come from the multispectral camera.
Hey, Richie, can you comment on how much time it took you to go through that effort of individually labeling pixels and how many roughly you had to label in order to train this?
Yeah, so it varies based on the mosaic that we were working with, but typically we would drop anywhere from 200 to 500 points on the image. And I would say that labeling process takes anywhere from 30 minutes to an hour roughly. Oftentimes your long wave infrared, near infrared, and red edge calibration procedures don't always yield absolute values. So we went with a supervised random forest classifier as opposed to an unsupervised method, just because our orthomosaic image values weren't always consistent amongst sites. So we sort of needed a specific classification procedure for each one of the orthomosaics that we developed.
Great, thanks, Richie. And so maybe that's a good transition into moving towards object-based identification instead of these pixel-based classifiers. So the project that we're working on right now, and as you can see, it's a very different landscape, but we are developing an object-based classifier in a regional convolutional neural network that is using both the shape of the object as well as some of the pixel data in order to classify different objects. So this has primarily been work that we've been doing in Southeast Florida. So this is right around Boynton Beach. And oh, none of my videos are going to play. But essentially what this CNN is able to do is identify different objects throughout this coastal imagery. So I think right now, Steve has it trained to pull out things like sea turtles, manta rays, sharks, humans, and also boats. So a group would be able to go out and fly the coast and feed their imagery, their raw imagery, into this classifier, and then pull out things like the geolocation or frame wherever these desired characteristics have been identified. And what we are working on now is adapting this for some of the work that we're doing up here in the Columbia River Basin to help with the identification of things like wood in the river, or even moving towards the ability to identify reds from aerial imagery as well.
So Sarah, question came in. So what data is the object-based detector using to determine the match, and how are you getting those values of like 99% match?
Yeah, that's a great question. And so the model is using both the arrangement of the pixels as well as characteristics like the pixel color in order to help identify these objects. And the way it works is that there's a moving window that moves across your image and basically does a binary conversion of, is this an animal, yes or no? And it's interesting, we've never seen 100% accuracy, so 99 is the best we get, but as that moving window moves through the image, it will essentially produce some level of uncertainty of whether or not that object is X. And so we've been able to train, as I mentioned, for a couple different types of objects. So in this example, we've trained for sea turtles, mantas, sharks, things that you see commonly throughout the coast. And you could do the same thing moving into these different environments. So now that we have that infrastructure built, we essentially all we need to do is swap out the training data. And it's a very similar process to what Richie was describing with the supervised random forest classifier, where we will sit through this imagery, and as that moving window is pulling out different parts of the image, somebody will need to train it to say, yes, that's a red, that's a tree, things like that. So it's the same general idea, but with an added layer of complexity, which is the shape and arrangement of those pixels. Okay, so that just about covers most of our efforts towards increasing the efficiency of our data collection tools. And at this point, I'm going to turn it over to Richie to talk about some of the analyses that we have been working on as well.
Thanks, Sarah. So some of your more traditional, what we would consider fish habitat relationships or aquatic habitat modeling approaches rely fairly heavily on what we would call suitability curves. So here's a few examples of what those suitability curves might look like for Chinook salmon for juvenile and spawning. And typically these curves are only built for depth and velocity, but oftentimes for spawning you might see a substrate curve as well. And these curves rely heavily on utilizing depths and velocities. So maybe you go out and measure depth and velocity in your stream, or in our case, we're fortunate enough to have extremely large bathymetric LiDAR data sets. So this is green spectrum LiDAR that can penetrate water and map the bathymetry of the river as well as the floodplain and any valley confinement features. We then develop a fully two-dimensional hydrodynamic model where we can model depths and velocities through the entire coverage of the bathymetric LiDAR, and we then run those depth and velocity outputs through these suitability indices to get an idea of how suitable the stream might be for a given species or a given life stage. And in this case, these HSI curves were developed specifically for the upper salmon, or a watershed in Idaho where we're doing this analysis. So we have watershed specific suitability curves that came from empirical data. So one really good way to visualize habitat suitability is to first take your depth suitabilities at each pixel and your velocity suitabilities at each pixel, and where they overlap, you calculate a geometric mean to get a single suitability value at each individual modeled pixel. You can then roll those up to what we would sort of consider a reach scale. So each one of these reaches is roughly 6 to 8 kilometers. That's the spatial scale they represent, and you can calculate a mean composite suitability across each reach to sort of get an idea of how the habitat might be changing through your watershed or through your site by species and by life stage. And this is sort of the most coarse analysis that you can do with HSI, but it's a good first cut just to sort of understand what life stage might be limiting and for what species and at what reach it might be most limiting. Another really good way to visualize these composite suitabilities is violin plots, and these violin plots can essentially be viewed as a frequency distribution or a histogram kind of flipped on its head. So you could imagine that your composite suitability values are between 0 and 1, 1 being completely suitable or the best possible habitat, and 0 being completely unsuitable. In this case, we're looking at those same 8 reaches, and this comes from the Lemhi River in eastern Idaho. It's just an example of some of these analyses that we've done for HSI. And in here, you can start to make some inferences about what species and what life stage is most limiting. So what you would want to note here is that for Chinook spawning, we have a large number of cells that fall above 0.75, indicating that the habitat is high quality spawning habitat. But we see the majority of the cells for juvenile summer rearing and juvenile winter rearing for Chinook falling below 0.25. So this is a result that we communicate to basin practitioners and restoration practitioners and engineers that if they're going to do a project on the ground, the type of actions that they take would most likely need to benefit juvenile rearing because we see that we have a lot of high quality adult spawning habitat. And then you can also look at how the geomorphology of the system might be impacting those suitability values. So that plot on the bottom is just looking at the percent of the geomorphic tier within each one of these reaches. And then the plot in the middle is steelhead, and what you'll note there is that primarily the habitat is quite suitable for steelhead spawning, rearing, winter and summer. So it's just a nice way to visualize the quality of your habitat through your system. A couple of the key metrics or common metrics that people like to calculate at a reach scale when you have this HSI information available is what we call the weighted usable area and the hydraulic habitat suitability. So weighted usable area is simply a summation of all of your geometric mean values across each reach. And you could imagine that as you add more water to the channel or as you move downstream in your system and you're increasing your wetted area, your weighted usable area is also going to increase. So we normalize it by the total wetted area of the reach, which gives us a hydraulic habitat suitability. And typically hydraulic habitat suitability values are again between 0 and 1. And what that tells you is really an indication of the quality of your habitat. So weighted usable area is going to tell you how much suitable habitat you have, but hydraulic habitat suitability is really going to give you an indication of the quality of habitat. So these blue bars are showing the hydraulic habitat suitability or the normalized weighted usable area, and the dots are showing the weighted usable area. And it's just another way to visualize and assess your reach or your site. And again, similar results as the last plots, we see that typically for juvenile summer rearing and juvenile winter rearing in this site in the Lemhi, you have very low hydraulic habitat suitability, so very low quality habitat, and you have very low weighted usable area, so not very much quality habitat. And again, for steelhead, we see much higher values across the board for HHS and weighted usable area. And these are the types of analyses that we can take, again, to folks working in watersheds and help them better direct their restoration efforts or what types of actions they might be putting on the ground. And I think I'm going to turn it over to Kevin now.
Yes, so I'm going to try to there we go. All right, so in addition to those HSI curves, we've also worked on developing other fish habitat relationships that have been based on empirical data collected at paired or sites where we had paired fish data and paired habitat data. So, these 200 or so meter sites sampled for fish, you get an estimate of abundance and density there, and also collect a lot of different habitat metrics. And what we're interested primarily in doing is pulling out what fish habitat relationships there, what about that habitat is helping to define the carrying capacity at those sites. And just to give you an idea of where those sites are or what that data set looks like, here's a little map showing all the sites in the Columbia Basin where we collected some of this data. You can see it's spread out across a variety of habitats, variety of watersheds. There's about 300 sites in the Chinook domain and about double that for steelhead. And so there's, as I mentioned, quite a variety of habitat, and we want to look at that empirical data and try to extract those fish habitat relationships directly from that in this exercise. So to do that, we're going to use a quantile random forest model. And I was going to take a couple minutes and talk to you about what some of that means and why I think that's an appropriate choice. So first, I just wanted to talk about why quantile regression can be useful, especially when thinking about carrying capacity. So in this example, if you had a particular habitat metric that was defining your carrying capacity and you measured it really well and you measured your fish density really well, and that relationship explained everything about how many fish you saw out there, you'd end up with a plot like that top one, a really nice tight relationship around that red line being your fish habitat relationship. Usually when you go out in the field, there are other things that you don't think of or can't measure that might be pushing some of those dots off of the line. For instance, you've got a big bull trout sitting at the bottom of your site that's chowing down on your small juvenile fish. You didn't see that bull trout, you don't know it's there, but it's certainly depressing the observed densities that you see. Or in our case, if you're dealing with an endangered species, a lot of times you may have the spawning escapement, the adults that came back the year before had really low numbers, and so they just won't fill up that habitat with juveniles that year. Again, pressing those points away from that red line. So you often end up with data sets that look like that bottom plot, more of a wedge-shaped distribution where there's things that you didn't measure or haven't thought of that are impacting the relationship you can see from that line, but we're still interested in pulling out that red line from a data set like that. And that's where quantile regression comes in. So you can fit quantile linear regression. You can basically fit different quantiles to that data set. And this plot over here is showing one example of that, where you're looking at winter densities in pools and comparing that to how much fish cover from large wood was available in that pool. And so those dots are the actual data, and you can fit a variety of quantiles to that. And if you can see the green line kind of running through, well, the middle one there, that's the 50th quantile. It's similar to the mean, very similar to your standard linear regression. And if you looked at that, you'd say, okay, there's a slight positive relationship. More large wood leads to slightly higher fish densities. However, that might not be how it's defining carrying capacity. You think about how many fish you might be able to put into that pool. And that might be more closely represented by the orange line, the 90th quantile, showing a much stronger positive relationship. And that's the kind of thing we're interested in pulling out when we're thinking about what about the habitat is defining the carrying capacity of those salmonid populations. So this is using quantile linear regression. You can do something similar with a random forest framework. And I don't know how many of you are familiar with random forests, but I'll talk a little bit about what that means. But essentially, it's a little more flexible framework. It's a little more data hungry. But the forest is essentially built of these regression trees. So in that top right plot, each of those little Plinko boxes or Plinko games essentially is a tree where you feed your habitat characteristics into that. And it's building some breakpoints along the way saying if your large wood is greater than 20 meters per some area, follow this right-hand branch. If it's less than that, follow the left-hand branch. And then it makes another breakpoint saying, great, if your substrate is more than 65% gravel, follow the right-hand branch. And if it's less than that, follow the other branch. And so for any particular site, right, where you have a fixed, you know what the habitat metrics are there. So you've got an X vector of inputs. You can feed that through each of these trees, and for that site, you'll end up at one of those little red circles, and that's your predicted fish density for that site. And when you put this together, you build maybe 1,000 of these trees, that becomes the random forest. That becomes a very robust predictor of whatever you're measuring, in our case, fish density. And so for a particular site, you end up with one prediction for each of those trees. You put them together, you get a distribution of fish densities, like that gray density plot down on the bottom right. And again, you could pull out the average value there, some of the mean or the median, about the blue or the green line there, which might be a typical analysis. In our case, since we are interested in carrying capacity, we want to look at that higher quantile. So maybe that red line there and say, I think this is what the carrying capacity for that site is based on those habitat characteristics. Another way to look at some of the results from that, and another reason why I think random forests are a great framework for this kind of analysis, is that it deals with nonlinear relationships really well. So you can imagine there's probably a lot of nonlinear fish habitat relationships out there. We'd like to express that. For instance, you can look at some of these. So these plots are showing the marginal effect of each habitat metric on our predicted carrying capacity. So assuming that everything else stays the same, if you look at the top left plot, our wetted width, as your wetted width increases, your fish per meter capacity also increases up to a point and it starts to level out. You can see other things like a couple plots over, the average August temperature. So assuming everything else stays the same, as your temperature increases, your capacity starts to drop. And so you can look at this as, again, the marginal effect. So it's not the entire model predictions, but it gives you some idea of what fish habitat relationships is that model pulling out. And it incorporates, as I mentioned, both nonlinear relationships, but also some of the correlations between your habitat metrics. And as you can imagine, for characterizing streams, a lot of the things about that stream are correlated to one degree or another. So this framework deals with that in a very natural way.
Hey, Kevin, had a question come in about how you selected your habitat metrics for the quantile random forest model.
That is a great question. Our data set, again, somebody mentioned it comes from the Columbia Habitat Monitoring Program, the CHAMP program, which collected hundreds of habitat metrics. And so we did try to winnow that down a little bit. We tried to capture a handful of metrics for different pieces of the habitat. So looking at things like, tell me something about the wood in the stream, tell me something about the substrate, tell me something about the size or the complexity of it. And we used the a maximum informational criteria, which is essentially like a nonlinear correlation coefficient, to look at which of those habitat metrics seem to have the most information about fish in each of those groups, and pull those together, or pull those out and put them into this quantile random forest model. So there's certainly some art to choosing those metrics, and we have developed a couple different flavors of these models, depending on what your question of interest might be. For instance, you can develop one group of metrics that might be more amenable to restoration, so more likely to contain things that you could change directly through a restoration design. You might have other flavors for answering different kinds of questions as well. But that's a great question. So then I wanted to get into talking about whether this works or not. And one of the challenges with that is when you're talking about carrying capacity, especially when you're looking at an endangered species like the Chinook salmon or steelhead, you often don't observe those sites at carrying capacity. That's why they're endangered. And so it's sometimes hard to validate those estimates of carrying capacity. This is one example, just the eye test. This site over here on the left has an estimated pretty high capacity for Chinook juveniles. And my fishery biologist friends tell me that looks like a wonderful place to find those fish. Similarly, the site on the right has a fairly low predicted carrying capacity for Chinook. That does not look like a great Chinook stream. I will say that that stream on the right does have a relatively high capacity for steelhead, and that looks a little more like a steelhead stream. So I think in that sense, this is passing the eye test. We also tried to validate it using some spawner recruit data from places where we could get it. So this is looking at one example. It's in the Chihuahua River in the Wenatchee River Basin in eastern Washington. And it's got a 30-year time series of spawners, so the adults coming back, and those juvenile par in the summer. And you can see all those points there in the plot, and you can fit your favorite spawner recruit curve to that. This is sort of the gold standard in fisheries of how you estimate carrying capacity, whether it's Beverton Holt or Ricker or a hockey stick. And you can see those curves, and the dashed line represent the corresponding capacity estimates for each of those curves. And you can compare that with the estimate from our QRF model, which is that red line. And you can see that dashed line falls right in the thick of these other ones. And the curve for those the QRF curve, I fit a Beverton Holt curve fixing the capacity parameter to what our estimate was. And what I want to point out about this validation is that it's two very different data sets. The spawner recruits is a long time series based solely on fish data. The QRF estimate at this point is really based on the habitat that you measured out there recently, and the fact that they're driving towards the same answer is very encouraging. So we did this for a variety of places around the Columbia River, mostly where we could get screw trap data with spawner estimates as well. It's a lot of plots here, but mostly I just want you to look and notice that the red lines are right in the mix with all the other curves. Again, two very different ways of estimating carrying capacity. The fact that they're all coming to similar conclusions, I think, is great validation for our QRF approach. One more way to look at it, just notice that the uncertainty, there's a lot of overlap, I guess, between confidence intervals in each method. And so that's great. We feel pretty good that it's working. We talked to you a little bit about what we can do with that. I mentioned one of the reasons we want to look at carrying capacity is we think that that's being defined by the habitat on the landscape. And it can be easier and cheaper and faster to measure the habitat than to try to measure sometimes the fish response. In particular, you want to look at those spawner recruit curves, you might need a 20-year time series to get a decent estimate of carrying capacity from that spawner recruit curve. However, you can go out and measure the habitat and immediately have some estimate of what that capacity might be. So this is an example of looking at in the Pacimmeroy, another river system in Idaho, where that black bar is our estimate of available summer par capacity for Chinook salmon. And the green bars correspond to data that's been collected over the last 10 years or so, and sort of the dark green is the average summer par that we've seen out there for those 10 years, and the light green is sort of the maximum that we've seen. So you can see, first of all, that average is about measuring up with the black bar, suggesting that summer par capacity might be one of the limiting factors in this watershed, kind of bumping up against the threshold that you might be able to get, how many fish you might be able to get out of this watershed. The blue bars correspond to recovery goals. So the MAT is the mean abundance threshold to hit recovery. And the dark blue bar would be the number of juveniles we think you need to produce from the Pacimmeroy in order to get back the number of adults that would hit your recovery goal. And then the light blue bar just adds 25% to that, because there's clearly a lot of uncertainty in many of these things, and we're interested in giving a little buffer and saying, look, let's set our goals a little bit higher than that mean abundance threshold, just in case. And so the difference between that light blue bar and the black bar is essentially the capacity deficit in that watershed. In order to meet recovery, we're going to need to increase that black bar to improve the capacity until it gets up to that light blue bar. And so there are lots of ways, well, some ways you might be able to do that. many different habitat restoration techniques and ideas, and this QRF model could be one way to compare different approaches and determine which ones would get you up to that light blue bar. This slide just shows how you can put those capacity estimates in context with some of those high resolution imagery that we were showing earlier. So these are showing 200-meter reaches and the estimates of capacity at those in conjunction with that high-res imagery. And you can see the dark areas are the high capacity reaches. And this is one more way to communicate our results to either restoration designers or general audience or other stakeholders. The idea being, take a look at some of these high capacity reaches, and you can see some of those in the insets and these maps. We'd like to make more habitat that looks like that. And sometimes that's easier for somebody to take in than to say, oh, well, I'd really like the large wood frequency to be greater than 12.3, and your percent gravel should be larger than 70%. And I mean, those are also useful, but this kind of helps bring that all in a holistic way. And hopefully by providing all of that information, it can really drive home what we're looking for in terms of a potential restoration design. The last part about this that I wanted to point out was just that it's a very scalable approach. So it can scale down as small as the channel unit potentially, and scale up to the 200-meter reach or up to the 5-kilometer reach, and then be extrapolated to the entire watershed. And depending on what your question of interest might be, one of those scales might be more appropriate at any point. So for instance, you might be focused at the watershed scale to say, well, I wonder what's the limiting life stage? Like that example in the Passamaquoddy, we conducted that similar analysis for a variety of watersheds in the Upper Salmon. And you can look at different life stages and figure out which one seems to have the most limiting capacity. Say, okay, I think this watershed seems to be limiting in summer rearing. Let's go focus on that. And then get into smaller scale, maybe that 5-kilometer scale, and look at the fish habitat relationships that we've pulled out and what areas may be most appropriate to try to apply some of those restoration techniques, and then inform what you might be trying to do with that restoration. What does a good pool look like? What does a nice 200-meter stretch of river with high capacity, what does that look like? Can we make more of those? Then you can also use this to examine pre and post restoration. So these two pictures are not exactly the same site, but it's the idea that you can measure the habitat beforehand, do some work, measure the habitat afterwards, and by estimating the carrying capacity before and after your restoration, look at the immediate impact you've had from that restoration work. And you do get that estimate immediately, as opposed to waiting for the fish signal, which could take years, if anything, because there might be some other factors downstream that you have no control over. But you can look and say, this is what we've done to the habitat right now and how much we've uplifted it.
Hey, Kevin, can I jump in for a second? We had a question about how you did the extrapolation, I think in the last slide, from the site all the way up to the watershed.
That's a great question. So the QRF model, let's go back to this one. When you have those direct measurements of habitat, you can make a direct estimate of the capacity. And so we had a number of sites scattered throughout the landscape where you can do that. And then there are lots of other places where you have what we're calling globally available attributes. So essentially GIS derived things like cumulative drainage area, maybe something about your predicted August temperature, your average annual discharge, other things like that you can get across the entire landscape. And we can use those as covariates in an extrapolation model, and then predict capacity at all of those points. And that's what you can sort of make out. The plot on the right is full of points, each of which represents about a kilometer reach. And those are all the predicted capacities based on that extrapolation model. with that, I was going to hand it back over to Richie to talk about leveraging legacy data.
Thanks, Kevin. Don't forget you're going to have to advance my slides for me. Having some bandwidth issues. So that was a really nice segue, actually, with the extrapolation model question. So go ahead and go to the next slide, Kevin. So, oh, too far. The current model is reliant on this Columbia Habitat Monitoring Program, which was a very intensive, labor-intensive, time-intensive program that looked at the habitat, measured the habitat anywhere from 200 to 600 meter reaches. And what that produced was a lot of these point estimates of the habitat across a bunch of watersheds. And we became increasingly reliant on a lot of these globally available attributes to then get a watershed scale capacity estimation. So go ahead and pop over to the next slide, Kevin. So this is what that extrapolation model might look like. Some of the globally available attributes or the continuous data that we utilize include some of these listed here. So temperature, growing degree days, a disturbance index, slope, channel classification. So we really wanted to come up with an approach that was less reliant on these globally available attributes to produce an assessment at a greater scale than sort of this 200 to 600 meter data that we were reliant on. And this CHAMP data, although a great program, it was roughly a decade of data collection. Oftentimes these tabular data are not as dynamic as we would like. So as machine learning comes online and becomes more readily available and easier to use, we wanted to come up with sort of a data permanence that would allow us to go back and revisit these data, you know, as the processing techniques improved. So again, we're moving towards a framework of data permanence, and this is really where the drone imagery comes into play. So imagery is very standardized. You can make some very simple rule sets about how imagery is collected in terms of front and side overlap, what types of cameras, what your ground sampling distance would be. There was a question about altitude, so you can set a lot of these rules for multiple agencies and multiple groups to collect imagery in a standardized way. That imagery can then be reposited into something like AWS, and it can be freely available for all of the groups to utilize. These data sources are very durable, and they provide a long-term sort of way of assessing status and trend monitoring across a much larger spatial scale than what was previously possible with past habitat monitoring programs. These data are also geo-referenced, so they're stored with their geolocation data, which makes it very nice for not only visualizing your data, but also for advanced analytics as we move into the future and we want to revisit these data. We can run them through these newly developed processing algorithms to produce new metrics that might be more important than what we could have measured on the ground previously. And so that's really all I wanted to touch on for leveraging legacy data. I'll
Richie,
turn it over to oh.
Before you move on, I had a question about the size of those imagery data sets. So say for a average kilometer of river, like how much information are you collecting and how do you store that size of information?
Yeah, that's a great question. So each one of these sites that we are sampling, if I'm referencing the Lemhi here, last summer we flew roughly 60 kilometers where we paired fisheries mark recapture abundance estimates with drone imagery and on-the-ground habitat data. We flew our MicaSense Altum 6-band camera, and that resulted in about 8 to 10 terabytes of raw imagery data, so not even including the processed orthomosaics. Currently, we house those on a local server, and we're moving towards more cloud-based solutions where we want to start putting our tools up on AWS or Azure, storing our data, and building a database that can be easily manipulated, read to, written to, and analyzed And so, yeah, these imagery data sets are very data heavy, but there are resources out there to really aid in the processing and storage of imagery data. And just to add to that, I mean, these resources are only getting better. So for us, it just made sense to try and get ahead of the curve right now, and start leveraging some of the technology out there, because in the future, I could imagine that the resolution of imagery that we're collecting from drones will probably be collected from satellites in the not too far off future.
All right, so this is just to sum up all of the things that we've talked about. And as Richie was talking about, we're really looking towards this development of a central repository where we can share a lot of the work that we have done. And that is a place where researchers can come and easily access data and evaluate in a more collaborative way all of the efforts that are taking place throughout the basin. So just to sum up some of the things that we talked about today, the DASH protocol, or the Drone Assisted Stream Habitat Protocol, is something that we are rolling out in a number of different watersheds as a way to reduce the cost of sampling, and then also our image processing tools that we are developing to reduce the time it takes to go and pull meaningful data out of those large imagery sets. And then, as Richie was mentioning, we're really working to make these a little more broadly applicable so that they're transferable to other data sets. And so not only is that data collection a scalable effort given the needs of the project, but we've built that ability to take in different types of data or different data detail into our modeling efforts as well. So a lot of the tools that we're developing on the analysis end are also scalable given the different data sources that may be available at these different projects. And then, like Richie was just talking about, our goal is to get at this data permanence that is setting up the infrastructure for this long-term status and trend monitoring, where despite the different flavor of projects that are happening throughout a basin or a watershed, we do have that raw data source, which would be the imagery that can be largely standardized, that produces this nice tool for long-term monitoring or for collaboration moving forward. So whether it be researchers in a watershed over or researchers in 15 years, we have this ability to go back and mine that imagery for whatever detail we need moving forward. And here I just want to point out Richie, Kevin, and then our data scientist, Mike Ackerman's GitHub, where a lot of these tools are publicly available. So please feel free to check those out. And then if you have any questions, we are always happy to get in contact with you and talk about some of these resources. And that's really all we have for you today. So we would be happy to open the floor to some discussion and take any questions that you all may have.
Excellent. Thank you guys for the presentation, and thanks everybody for attending the webinar. As Sarah mentioned, we're all going to hang around here for a little bit. So if any of you want to stick on the line and ask some questions or elaborate on some details, please feel free to.
Nice job, guys. Great information.
Did we do such a good job? There's no more questions.
Everybody's pondering to come back with more questions.
Good.
So I've got a point of elaboration maybe for Kevin. Kevin, can you speak to the ability to generate QRF estimates in locations where you have no on-the-ground data?
Sure. That would be part of that extrapolation model that I talked about. So places where you have detailed data, you're using some of those, whatever covariates we can get that are available everywhere. And we are constantly looking for new ones that might perform better or be more tied to capacity. So you're spreading that out across different watersheds that might, some of which may have that detailed habitat data in spots, other watersheds may not at all. And you can still do that. You can still extrapolate to those areas. And some of those spawner recruit validation curves were for places where we don't have any detailed habitat data, and yet it's still estimating capacity fairly well.
Excellent. Actually had a question roll in. I work with coho on the coast. How difficult is it to apply these tools to different species?
So I'll talk a little bit about the QRF would be, I don't know if I would certainly wouldn't want to apply a Chinook model to coho, right? But to fit a new model, I mean, as I mentioned, for our data set, we had about 300 sites for Chinook. It's paired fish habitat. So if you have something along those lines for coho, you certainly build a different QRF model for coho. The random forest type algorithms are a bit data hungry. Part of it's the trade-off with how flexible they are, but there might be other ways you might be able to just do a standard quantile regression type approach, linear regression, or something along those lines if your data set was smaller. But it does rely on that paired fish habitat data set. HSI curves, they must have HSI curves for coho. So if you have that sort of depth and velocity, anything else that you want to have a curve for on the coast, then you could certainly apply that for those areas. Richie or Sarah, do you guys have anything to add to that?
So maybe just to elaborate on that a little bit on the data collection side, I think that when we were looking at things like pulling data from the drone or from satellite, that really was an effort to figure out how we can get enough data to feed something like a QRF model in the future as we expand outside of the Columbia River Basin and outside of that CHAMP data set. So we really are looking at ways to either rely exclusively or more heavily on the drone or remote sensing for that habitat data. So then I think, as Kevin mentioned, it really goes back to having the fish data as well. So we really have been working towards moving this out into other areas. So just for example, something that we've been looking at is applying similar methods to something like sea turtles moving through a mangrove system. So we are trying to figure out how to make this more adaptable without that data-rich legacy data set.
All right, it looks like questions are pretty much done rolling in. You guys have any concluding remarks or anything you'd like to add?
Thanks to everybody who joined the call. Appreciate giving us the chance to talk to you about some of our work.
Hey, Chris, sorry, this is Elsa. Sorry to interrupt. We had a request on my personal chat for somebody to reshare the GitHub links, please. Thank you.
And Elsie, you may have mentioned this already, but it might be worth mentioning again where people can find this presentation if they want to go back and look at any of the slides.
The webinar recording will be emailed out to all registrants by tomorrow, and we will also post it on our website at biomark.com.
Excellent, thank you.
You guys have gotten some positive feedback, some thank yous as well, and some nice jobs. All right, Elsa, I think that most of the questions are done. Shall we go ahead and cut this off?
Yes, thank you.
Thanks, everyone.
Thanks.